Well, thanks very much for the invitation to present to you. Steve and I have been working, Steve and Katai and I have been working on developing this AOP for about a year now, and so I'm going to talk to you about the science that is used to incorporate into this AOP that's relevant for arsenic-induced developmental toxicity. And the data that we use to develop the AOP would not be possible without efforts from my collaborators in Mexico and graduate students, both current and former, from the lab. So I wanted to acknowledge their work. My lab really focuses on mechanisms that underlie toxic metal-induced disease, and we focus largely our efforts on inorganic arsenic, the king of poisons. So we know that arsenic continues to contaminate almost every continent on the globe, with areas like West Bengal and Bangladesh very severe global public health crisis. Work that comes out of our Superfund program at UNC has actually highlighted concerns for inorganic arsenic in private wells in North Carolina. We have 2 million people who are on private wells, and as many of you know, the issue with private wells is they're currently unregulated, and so many of these individuals may have levels that far exceed the EPA and World Health Organization standards. So for example, in our assessment, the highest level that we saw was 800 parts per billion, which is 80 times the EPA limit. So something that we have to be considering here as well as other countries. And our concern about this exposure is really related to the health effects that it can cause. So it's a group 1 carcinogen, so established tumorigenesis in skin, bladder, lung, liver, prostate, and kidney. And it's also associated with non-cancer endpoints, neurological disorders, reproductive effects, cardiovascular disease, and diabetes. So a whole slew of diseases that are associated with exposure. One of the outcomes that's really interesting and strongly reproducible across different cohorts is an association between prenatal exposure to arsenic and infants being born at lower birth weight. So this is a figure that comes from a recent meta-analysis published in EHP, which looks at published 4, and then since then 3 other studies, including ours, support that prenatal arsenic exposure reduces the weight of infants at birth. And this is of concern because being born at lower birth weight puts you at risk for diseases later in life. In fact, we know that from mouse models and from human populations that there is an established association between early life exposure and later in life health effects. So Mike Walkies has shown increased hepatocellular carcinomas in mice, and Alan Smith from Berkeley has shown increased cancer and non-cancer deaths associated with this early life exposure. So if you're interested in studying the developmental origins of health and diseases, arsenic really is a great model contaminant. And we in the toxicology field have been focused on trying to understand the mechanisms of inorganic arsenic for decades. This review from Mike Hughes describes at least 9 different ways that arsenic can induce damage that can be through reactive oxygen species, altering DNA repair capacity, modifying cell proliferation, as well as epigenetic modifications. But one of the gaps that still remains is really understanding which of these mechanisms is relevant to the developmental toxicity that we see in relationship to arsenic exposure. And furthermore, what we don't have necessarily a good handle on is this chain of events that links arsenic exposure to developmental toxicity. And so, like I said, about a year ago, Steve and Katai and I started brainstorming about ways that we could try to fill these gaps, and decided to begin to develop this AOP related to various things, and we're working on other AOPs as well. And as Steve said, this really is, this construct helps to portray linkages that are very useful in identifying gaps in knowledge. And one of the pieces that I really find valuable about developing the AOPs is being able to show this sequential chain of causally linked events. And you can bring in data sources from different studies. And so while this is, the writing and text is probably too hard to visualize, I wanted to share with you some aspects of the AOP that we're developing. And of course, these key components of the AOP include the adverse outcome. And in this case, the adverse outcome is the lower birth weight effect that I had spoken about before in relationship to arsenic exposure. And then we have key molecular effects and molecular initiating events. And so the graphic that you're seeing, the key events are indicated by the nodes, and those are really important because they reflect changes in biological state that could be measured and are essential for the progression of disease. And then we have the edges or the key event relationships, which are the connections between the two events, and that's where we begin to build our scientific basis for these connections. And so I'm going to talk about data that we've used to build the AOP at each of these places that I've indicated with a red arrow. And some of this comes from a study that I launched about 7 years ago in Gómez Palacio, Mexico. So this region of Mexico has elevated levels of arsenic in water, but no one had done a biomonitoring study there. So I worked with my collaborator, Gonzalo Garcia Vargas, who's pictured there on the left, recruited women who were pregnant, analyzed arsenic in their water, analyzed arsenic in their urine, and all of the methylated metabolites of arsenic. And what we found was that their drinking water ranged up to 240 parts per billion, which is 24 times the EPA standard, and found that more than half of the population was drinking levels of arsenic that are considered harmful. And so, of course, we provide this information back to the women, and this represents one of the major initiatives in my lab, which is to prevent the possible exposure during pregnancy of toxic metals. And so I described that effect of prenatal arsenic exposure on birth weight, and the graphic that you're looking at the right comes from our study. As you look from left to right, you see that arsenic exposure is increasing. As arsenic exposure increases, the birth weight of the infants decrease. Because of the data that comes from mouse models, which really showed that early life exposure is associated with later life disease, and because of evidence that Mike Waukes provided years ago looking at specific candidate genes and CpG methylation, we focused our assessment initially on methylation as a potential, CpG methylation as a potential mechanism controlling critical genes in pathways that may be linked to birth outcomes in infants. So we used the 450K Illumina array. We analyzed DNA methylation in the cord blood leukocytes of the infants in Mexico. We integrated the methylation with gene expression data, and then we also required that the methylation and gene expression was associated with a birth outcome. And when we did that, we found a set of really interesting genes, and this graphic shows you the pattern of one of these. This gene is called KCNQ1, and as KCNQ1 methylation increased, so as CpG methylation increased in KCNQ1, expression of KCNQ1 decreased, and that's one of the sort of generic relationships that we can see with methylation, that it can serve as a silencing mechanism. One of the reasons this gene is so interesting is that it's an imprinted gene. It is located on the short arm of chromosome 11. This area of the chromosome is a hotspot for imprinted genes. So you can see the coloring here. Pink represents maternally expressed alleles, blue represent paternally expressed alleles, and you can see that it's highly conserved across human and mice. And so the other interesting thing about, and KCNQ1 is a potassium channel protein, and its location is indicated with the red arrow. So an interesting feature about this area of the chromosome is that it appears to be a hotspot for insults that are occurring during the prenatal period. So a very famous study was published that looked at preconception exposure to famine during the Dutch hunger winter. And the investigators found that there was increased methylation of another gene that's not far from KCNQ1, insulin growth factor 2, and found that this methylation was actually associated with that exposure to famine during fetal development and associated with later life growth, later life obesity. So again, this area appears to be a hotspot for susceptibility to environmental insult that's occurring prenatally. So these imprinted genes are really fascinating. They don't follow Mendelian laws. Their expression is tied to the parent from whom the allele is inherited. And I love this picture that comes from a Nature article, which is this tug of war. So this is a hypothesis, in a way, for these genes that really control fetal growth and fetal size. And so the hypothesis for their existence is that it's a genomic tug of war between the mother and father and the maternal resources of the fetus, where maternally imprinted genes tend to be growth restricting, and paternally imprinted genes tend to be growth promoting. So depending on whether these genes are turned on or off, the size of the fetus varies. And so interestingly, the gene that we identify here to be associated with prenatal arsenic exposure, KCNQ1, is there are many studies that show that this gene is associated with fetal growth, not surprising because it's an imprinted gene, but also it's associated with later life growth and metabolism. I had mentioned that our methylation was done in cord blood, and that's because it was impossible to collect placentas at this study site in Mexico. And if I could have had placenta, I would have, because we actually believe that it's expression in the placenta that is likely driving fetal growth. There's a very interesting paper that Susan Murphy from Duke has published, where Susan looked at methylation of imprinted genes across different tissues, fetal tissues, and showed that if you look at methylation across imprinted genes and these tissues, they're highly conserved. This is because imprinted genes escape the methylation erasure that happens early after fertilization. So they're protected, highly stable, and highly conserved, which means that even though we didn't have placenta, the methylation marks that we're seeing in cord blood are likely strongly correlated to what we would see in the placenta. And so in terms of new science that's gained, we're understanding now a mechanism of action for arsenic's effect on fetal growth, which is likely linked to this gene. And so as we're building out the AOP, we have this organismal effect, this lower birth weight effect. The cells that we're looking at are likely, the cellular effect is happening in the placenta. And then the molecular event is this DNA methylation that's influencing gene expression. And so one of the questions that we're interested in answering now is, what is the mechanism by which arsenic targets KCNQ1? How is it that you're exposed to arsenic and you have these very specific genes that you see differentially methylated? Because if we look at the enzymes that are modifying cytosines, if we look at DNA methyltransferase, this enzyme really doesn't have a mechanism to be site-specific. And yet, if we look in data that come from human populations exposed to environmental contaminants, and I'll show you some data that we generated in a study a few years ago where we were looking at prenatal cadmium exposure. These infants actually come from North Carolina. So we were looking at prenatal cadmium exposure and asking, what are the genes that showed differential methylation? And we found 61 that were differentially methylated in relationship to cadmium. And when you look at those 61 genes, you really don't see a pattern until you start to look at the promoter sequences of these genes. And something really interesting turns up when you do that. So when you look at the promoter regions of these 61 genes, they are highly enriched, which means that across many of these genes, we see binding sites for a transcription factor known as MTF1. And MTF1 is a metal responsive transcription factor. So here's a transcription factor that we know responds to metal exposure. It's sensing metal exposure in the environment. And in these genes that show differential methylation, we see an enrichment for this transcription factor. And so that led to a hypothesis that first we have provided more data for recently, where we've looked across every available study of prenatal exposure to an environmental toxicant and CpG methylation, where that CpG methylation was analyzed in a genome-wide context. And so there are 14 studies that have been published to date. Those include our cadmium paper, so cadmium-induced methylation, as well as our analysis of arsenic-induced methylation. Now, when we look across these studies, one of the first questions that we wanted to ask was, do we see a commonality? If you looked across these studies, if you had more than one study that was focusing on arsenic. I forgot to ask for a pointer. Oh, here it is. So if you look here, we have four studies looking at arsenic exposure. And then each of these colors on the heat map indicates a gene. If it's red, it was hypermethylated in relationship to arsenic. If it was blue, it was hypomethylated. And then you can see we have 2 studies on cadmium, lead, manganese, mercury, and tobacco smoke. So you can see very little conservation at the gene level as we're looking across these studies. Now, if we ask again that same question, but what if we look at the promoter regions of these genes? Even though the genes aren't showing us similarity, even within a contaminant across study, what happens if we look in the promoter regions? And so again, we're seeing this very strong evidence for a role for transcription factors in driving the gene-specific methylation that we're looking at. And so the way that I visualize this hypothesis, when we're exposed to an environmental contaminant, we have transcription factors that are protecting us and responding in the cell. So here's our TF. So an example of that would be MTF1, right? We're exposed to metals, and metal responsive transcription factor responds and binds to its target genes. And when it does that, it would influence accessibility of that DNA to DNA methyltransferase. And that would influence whether or not that specific region of a gene is hyper or hypomethylated. And similarly, if we have inhibition of a transcription factor, now this area of the genome is accessible for DNA methyltransferase. And so we're working on this right now in the lab, where we're taking cells and driving transcription factors to be induced or repressed, and using genome-wide methylation to see whether or not we're seeing an enrichment for that methylation. So this is important because we really need to understand how it is that environmental contaminants are influencing these target genes. If we do understand that, we have the beginning of what we need for potential intervention strategies. So just coming back now to here, we had this molecular effect of the epigenetic alteration, and we have transcription factor modification as a likely molecular initiating event that's influencing that methylation patterning. So in summary, we really believe that the AOPs do provide a great framework for helping us to understand the relationships, but also a visualization and a way to look at the strength of the scientific evidence for these key components underlying, in this case, arsenic-induced low birth weight. And also helping us to integrate various data from human population-based data to cell culture in a really uniform way. So with that, I will acknowledge my collaborators again and happy to take any questions.